Hello and welcome back everyone to my channel. My name is Shirsha and in this particular series we are talking about mammalian cell culture. Now mammalian cell culture is a pretty vast topic and it is commonly practiced in almost all laboratories. So instead of covering it all at one go, we are covering it in a number of sections. In the previous video uploaded by me on this channel, we talked about the various equipments that are commonly used in mammalian cell culture and what is the utility of each of these equipments, what is the working principle, and so on and so forth. In this particular lecture, we are going to be talking about the various requirements for mammalian cell culture. That is, the various conditions that are required for maintaining a healthy culture. Now, I would like to reiterate once before we delve into the topic, the importance of mammalian cell culture. Now, for any experimental work that you're doing, you would always want to replicate your work, or you would rather always want your work to be valid in terms of living systems. In vitro experiments are the foundation of any further experiments, but once your experiments work in vitro, you would always want to see their validity in cellular context. Now, before you take your in vitro experiment to in vivo, that is where you'll be doing them in animal systems, the intermediate step will be to do these experiments in cellular systems, and that is where cell culture comes in. Mammalian cell culture is one of the most commonly practiced cell culture methods because over here it involves the utilization of a eukaryotic cellular system. So therefore, you can now validate your studies and your findings in vitro in cellular condition or in cellular context. So moving ahead, we are now going to start discussion about the various conditions for a healthy culture. So the first and foremost is maintaining the appropriate temperature. So as you all know, if you think about the human body, for example, then the human body is maintained at a specific temperature, and this temperature is very important for proper functioning of the entire body system, including the various enzymes and proteins in the body. Similarly, just like in humans, in other animals also there is an optimal body temperature at which the cells survive the best and they work the best. So maintaining the temperature is very important. Most mammalian cell cultures are grown in incubators that is set at 37 degrees centigrade, and this temperature is the same as our own body temperature. This is chosen because it is the core body temperature of Homo sapiens or humans. However, there is no reason to expect that 37 degrees centigrade would be optimal for all types of human cells, and certainly not for all cells from all mammals. So just like you cannot really say that 37 degrees centigrade is the optimal temperature for all types of human cells, so it is a big stretch to say that all human cells grow at 37, because if you think of our body, and I may deviate a little bit, but not the entire body is not maintained at 37. For instance, the testes reside outside of the body to help maintain a lower temperature. So just like not all human cells can grow at 37 degrees centigrade, not all mammalian cells can also grow at 37 degrees centigrade. But yes, it is helpful to keep in mind that most of the different cell lines that we use for cell culture, if these are continuous cell lines which are already established, then these would typically be derived from humans themselves. But they can be derived from other animals as well. For instance, in case of insect cells, for example, we will not be growing them at 37 degrees centigrade, but rather we will grow them at 27 degrees centigrade. Although it is not really mammalian cell culture, but that was just to give you an example that different cells thrive best at different temperatures. However, if the line has been in common use for many years, then the cells might be best adapted to this temperature as a result of continued adaptation. Insect, worm and fish cells, on the other hand, cannot be grown at 37 degrees centigrade and require significantly lower temperatures, like I already mentioned. The primary role of maintaining temperature in cell incubators is of the incubator. So the CO2 incubator that we talked about in our last lecture, that plays the main role in maintaining the temperature. And not only does it maintain the temperature, it also helps maintain the humidity and the required level of gases, carbon dioxide and oxygen. The next condition that we are going to discuss about is pH. So just like different enzymes and proteins have an optimal temperature at which they function the best, similarly they also have an optimum pH at which they function the best. And in order for cells to survive and be healthy, not only is it important to maintain the temperature, but it is equally important to maintain the pH of the media in which the cells are growing. Regulation of extracellular and intracellular pH is also essential for survival of mammalian cells. The pH is not only important for maintaining the appropriate ion balance, and mind you, maintaining the ion balance is also very critical for survival, but also for maintaining the optimal function of cellular enzymes and for optimal binding of hormones and growth factors to cell surface receptors. So a cell is a living entity, and in order for a cell to thrive and to survive, all of its enzymes need to be operational, and they would be functional only at their optimal pH and optimal temperature range. So yes, do keep note that it is not that you have to maintain only a single value for pH or temperature. That is, the temperatures and pHs are not restricted to just a single value, but there is an allowed range within which the cells would survive the best. Now, if you have studied about enzymes, you know that for enzymes as well, there is a pH range as well as a temperature range at which they function the best. And if one were to plot the pH versus activity curve of an enzyme, or let's say temperature versus activity curve of an enzyme. So let us assume that we have the activity on this particular axis, the y-axis, and the pH or temperature on the x-axis. Then the curve that it would give would somewhat be a bell-shaped curve, where at this point, that is at this particular pH or temperature, you have the best activity, and then within this particular range you get the activity of the enzyme, and beyond that, if you increase or decrease the temperature and pH any further, then the activity of your enzyme will go down drastically. So similar to this, or rather as a result of this, for healthy survival of cells and for healthy proliferation of cells, maintaining pH is very essential. Even transient changes in pH can alter cellular metabolism and induce the production of heat shock proteins, a process which can eventually lead to the apoptotic death of cells. Most media strive to achieve and maintain a pH between 7 and 7.4, and that is the pH which is maintained in our bodies as well, with a median of 7.2. Cells differ widely in their ability to tolerate significant deviations from this level. Some cells which are more resilient, they can tolerate wider deviations, but others which are more sensitive, they would be affected by even the slightest of deviations, and even the slightest deviations can lead to their death. Most cells will tolerate a medium in the range of 6.5 to 7.8, but media much outside this range can end up being lethal for the cells. Now, the regulation of pH is done using a variety of buffering systems that are integrated into the media, that is, that are a part of the media itself. As with temperature, slow changes in pH are tolerated better than rapid changes that cause apoptotic cell death. So this is true for any kind of change. Gradual changes would be better tolerated. However, if you suddenly change the condition, then these would be more stressful for the cells, and the cells would eventually end up dying. All cells produce lactic acid and carbon dioxide as a byproduct of their energy metabolism. So as cells metabolize, they produce CO2 as well as lactic acid, and both of these compounds actually affect the pH of the medium. How? They make the pH more acidic. Lactic acid itself can be taken up and metabolized by some of the cells, but not all of the lactic acid produced by the cells as a result of aerobic respiration would be utilized. Additionally, the CO2 produced by the cells will affect the pH through the bicarbonate carbon dioxide buffering system. So the bicarbonate carbon dioxide buffering system is the most commonly used buffering systems for mammalian cell cultures, and we will be discussing about it in much more detail in the upcoming slides. So the CO2 that is produced by the cells as the cells respire, they will get integrated into and become a part of this bicarbonate CO2 buffering system, and the rest of it would be released into the atmosphere of the culture container. That is, if you recall the petri dishes or the flasks or the flat flask or the conical flask that I showed you in the previous lecture, then the rest of the CO2, that which does not become a part of this buffering system, will be released into the empty spaces of the culture container, and it will keep on affecting the pH of the media by continuously getting integrated into the buffering system. Now, the actual pH in a given culture at a time is therefore determined by a number of factors, which include the culture configuration, the medium that the cells are grown in, the cell type, as well as the buffering capacity of the media. So pH is not regulated by one, but by multiple factors. The main buffering system in most media is the carbon dioxide bicarbonate buffering system. The interaction of CO2 derived from the cells or the atmosphere with water leads to a drop in pH as described by the equation below. So CO2 is produced by the cells as they respire and it is released into the media. So CO2 from the cells and the CO2 from the atmosphere. By atmosphere, I mean the atmosphere within the CO2 incubator will interact with water to produce H plus and CO3 2 minus. So you get protons and you get your carbonate ions. And these protons will lead to a drop in the pH, because pH is ultimately, or pH of a solution is ultimately a measure of the availability of free protons in that solution. Higher the concentration of free protons in a solution, lower is said to be the pH. So therefore, these protons produced as a result of the reaction between CO2 and water leads to a drop in the pH. But the buffering system has another component, the bicarbonate. So sodium bicarbonate is one of the common ingredients of the cell culture media. Increasing the bicarbonate concentration neutralizes the effect of increased carbon dioxide. How? So you have your sodium bicarbonate that dissociates to form sodium ions and bicarbonate ions. Now, whatever free protons would be produced in the solution as a result of this particular equation, these protons will combine with this bicarbonate produced by the dissociation of NaHCO3 to give rise to the formation of H2CO3. And as that happens, these protons that are being produced in the solution will get used up, and therefore the availability of free protons in the solution will go down and the pH will be prevented from decreasing. So the increased proton ion concentration drives the equation to the left until equilibrium is reached at pH 7.4. Now if the correct bicarbonate CO2 ratios are used, obviously you can't really control the level of CO2. What you can indeed control is the amount of bicarbonate that you're adding to the media while preparing it. So if you add the correct bicarbonate and if your incubator is maintaining the cells at 5% CO2 level, then at equilibrium, the pH should be around 7.4. High concentrations of bicarbonate in media formulations would require higher CO2 percentages in the air supply to provide an appropriate pH in the medium, but also to provide greater buffering capacity. So therefore, it is very, very important to strike a balance between the amount of bicarbonate that you're adding to the medium while preparing it and the level of CO2 that is maintained in the incubator, because these two will interplay among themselves to determine the pH of the medium and also the buffering capacity of the medium. What do we mean by buffering capacity? Buffering capacity of the medium refers to the ability of the medium to maintain a particular pH despite certain fluctuations in the level of protons. Now, the carbonate ions themselves will also affect cell function to some extent, and some cell types require bicarbonate to grow independent of its requirement as a buffer. So, under such situations, the amount of carbonate that you would supply in the medium would be greater, because some amount of this carbonate or bicarbonate will get used up by these cells for their normal survival, and therefore lesser amount will be available to act as a part of this buffering system. The bicarbonate buffering system, while inexpensive and well understood, may be supplemented with a variety of organic buffers. So yes, a variety of organic buffers are also available, which you can use to supplement this bicarbonate CO2 buffering system. And typically it will depend upon your cell type if you are going to resort to these other organic buffers. For most of the commonly used cell lines like HK cell lines, which is the human embryonic kidney cell lines, you can use just the bicarbonate CO2 buffering system, whereas use of other sophisticated cell lines, for instance like hybridoma cell lines, require the use of specific medium which already come with a buffering system. You don't really prepare these medias, you are not supplementing them with the bicarbonate ions, and therefore you're not really providing this bicarbonate CO2 buffering system, but they come with other more sophisticated buffering systems that are obviously not disclosed to the public for trademark purposes. Sorry, not trademark, but for company secrets. uh, Anyways, we are deviating. So the most widely used bitter ionic buffer is HEPAs. The next condition that we are going to be talking about is the osmolarity. Now, osmolarity is a measure of solute concentration defined as the number of osmoles of solute per liter of solution. The osmolarity of the medium used is determined by the medium formulation. Salts and glucose are the major contributors to the osmolarity of the medium, although amino acids presence may also contribute significantly. The next condition is the gaseous content. Now, the CO2 level is generally a part of the buffering system of the medium and must be therefore set accordingly. So, the CO2 level set will depend on the amount of sodium bicarbonate that you're adding in the media formulation. Higher the amount of sodium bicarbonate you're adding, higher would be the CO2 level. But then again, there are optimal ratios which have to be adhered to ensure best survival of the cell and best pH maintenance. Oxygen levels in the incubator are provided by the 90 to 95% of air in the incubator mix. So the CO2 incubators, as we have already discussed in our previous lecture, is hooked up to these carbon dioxide cylinders, and the CO2 level within the incubator is typically maintained at 5%. So the rest 95% is your environmental or atmospheric air that is present within the incubator, and this will provide the desired oxygen levels or the required oxygen levels. This means that the PO2 in most incubators is around 18%. So if you calculate the partial oxygen pressure within the incubators, that will turn out to be somewhere around 18%, which is very similar to the kind of PO2 that cells face in the body system. So the next requirement for cell culture is media. Now, media is one of the, well, I can't really say which of these is the most crucial, because all of the conditions that we have discussed so far are equally important for maintaining healthy cells in cell culture and for successful maintenance of your cells. So another important requirement for cell culture is media. The pioneers of cell culture used exceedingly complex environments to maintain their pieces of tissue or cells in vitro. including plasma clots and tissue extracts in complex hand-blown glass chambers designed to maintain sterility whilst providing gas exchange. Then, with the use of better and more complex media, serum supplementation alone was sufficient to grow many transformed cell types in culture. However, there are still some other cells which require a feeder layer of other cells that were treated so as to be incapable of proliferation but provided the necessary nutrients or substrates. So in case of feeder cell layers, what you do is that you have your cell culture dish. At the bottom surface or at the bottom, you have these layer of feeder cells. Now these feeder cells have been treated so that they themselves do not multiply, and therefore they will not be utilizing much of the nutrients of the medium provided for their own growth, but rather as these cells metabolize, so they will not increase in number, but they will still be alive. So as they metabolize, they will be producing certain components which will be required by these other cells for their growth. So these feeder cells become essential for the survival of these specific cell types. Now, the culture media required for the propagation of animal cells are much more complex than the minimal media sufficient to support the growth of bacteria and yeast. The choice of medium is still often quite empirical. You can't really use the same media for different cell types. So it is very, very important that you read the literature and determine which medium has been used previously for a particular cell line. However, I would like to mention over here that reading the literature is not always good enough, and sometimes you might not, or the medium which has been used in literature or which has been reported in literature might not work in your hands, and it might require some optimization in your hands while you're practicing. So yes, be very cautious when selecting the medium. Do a thorough literature review to see in what kind of media those particular cells that you're going to be working with have been previously grown, and in which medium these other experiments have been done. That will give you a guideline as to where to begin from. If several different media have been used, and this is often the case, they may all have to be tested to see which of them work best at your hands for your specific experiments. Once a type of medium has been selected, it should be kept consistent for as long as possible. Although continuous cell lines can be accustomed to grow on different media as long as the conditions are constant. But yes, within your laboratory, when you're working with a particular cell line, try to not change the medium in which the cell line is grown. Try to maintain the medium as constant, because this would be best for your cell's survival and for your cell's health. Now, all media are composed primarily of water. So just like the human body is 70% water, and water is an integral component of cells which is required for their normal functioning. So all media are composed primarily of water. If media is prepared in the laboratory, then the source of water can actually make a huge difference in the quality of the media being prepared. Ideally, you should always use autoclaved double distilled molecule water. The medium provides essential nutrients that are incorporated into dividing cells, such as amino acids, fatty acids, sugars, trace elements, vitamins, cofactors, and ions and molecules necessary to maintain the proper chemical environment of the cell. So as you can figure out from this long list, that medium required for mammalian cell culture are quite complicated concoctions. Additionally, the medium also contains all or part of the buffering system. Like for instance, if we think about the bicarbonate CO2 buffering system, then the medium contains just a part of it, the bicarbonate part. So the medium contains all or part of the buffering system required to maintain a physiological pH and should provide the appropriate osmolarity to the cells as well. So typically this is what your medium would look like. This is Dalbeco's modified Eagle medium, also commonly abbreviated as DMEM, which is one of the most popularly and commonly used mediums in animal cell cultures all over the world. So this media is red in color, and this red coloration is given by phenol red, which also acts as a pH indicator. So this will allow you to simply gauge the pH of your media by visual inspection only. So this goes on to show how important pH is for the health of the cells, and any change in pH can affect the growth of cells. So if you see that your pH is starting to turn slightly orange and yellow orange, that means that it is becoming acidic. So it means that you should either change the media or you should subculture the cells. Or if you see that the media has gone completely yellow, then it is quite likely that your cells have started to die already and they will not be appearing very healthy if you observe them under the microscope. So here we have a comprehensive chart discussing about some of the most commonly used mediums, their type and their description. So minimum essential medium, or MEM, was the precursor of DMEM. It's a basal media. It is used for primary and diploid cultures. It contains only vitamins, non-essential amino acids, inorganic salts and glutamine. Then we have DMEM, Dalbeco's modified eagle medium, which is one of the most commonly used cell culture media. It is similar to MEM, but what are the modifications? It contains additional amino acids and vitamins, and it does not contain any growth promoters or protein. Now we have these other modifications called IMDM, or Iscove's modified Dalbeco's media. Now this is a complex media which is essentially a modified version of DMEM, modified to contain more amino acids and vitamins. It contains no iron but does have selenium and ferric nitrate. So some ions are also included in it, and it is suitable for high density cultures. Next, we have the Rpmi medium. Now, Rpmi medium is also another complex medium that works for most types of mammalian cells, and it is one of the most commonly used medium. And finally, well, this is not the end of the list. The list goes on and on, but these are just the 5 different kinds that we are going to discuss for the sake of example. We have Ham's F10 and F12 medium. Now these are serum free media. So Ham's F10 was the original ham nutrient mixture designed for use with the CHO cells, that is the Chinese hamster ovary cells. It is suitable for use with human diploid cells. Ham's F12 is more complex and is suited for rat prostate epithelial and rat hepatocyte cells. Now this is just information, and what medium you have to use will depend on what cell type you're using, and it will depend on that cell type's history. Like if the previous workers of your lab have already been working with that particular cell type, then what medium were they using for that cell type? Or if you have gotten that cell type from a collaborator, then what media were the collaborators using when they were working with that cell type? Or if you simply commercially purchased a particular cell type, then what is the suggested medium for growth? So depending on that, you will select your specific media. So some components may perform more than one role. For example, sodium bicarbonate may be used as a carbonate source for the cells, but may also play an important role in maintaining the appropriate pH and osmolarity. So it is not necessary that a particular component of a media has only one role, but its roles can be multifaceted. Amino acids and glucose, as well as ions such as sodium chloride, contribute to the osmolarity of the medium as well as have nutritional roles. In addition to the bicarbonate carbon dioxide buffering system, the medium may also contain some phosphate buffer and some complex organic buffers to help maintain the ideal pH. Media may also contain antioxidants or other reducing agents. Now, all of these will depend on your specific cell type. Again, everything comes down to your cell type. What you're going to add to your media, what you're not going to add, it all depends on the cell's specific requirements. Most media contain phenol red as a pH indicator, which gives them the red color like we already discussed previously. This is helpful in rapidly assessing the pH of the medium of all the cultures kept in an incubator. Most media, for example, minimal essential media or the Dalbeco's modified eagle medium, were developed specifically for use with serum supplementation and high density growth of cells. So what is serum? Serum is a part of the blood, as you all know. Serum is basically the blood plasma without the clotting factors. So if you remove the blood cells and the clotting factors from blood, what you're left behind with is called the serum. And most of these media like MEM and DMEM were developed to use or for use along with serum supplementation, because it will provide some essential factors and components essential for the cell's growth and survival. Other mediums, such as Ham's nutrient medium fixtures like F12 and F10, were tailored specifically for growing a given cell type at low density with a minimal amount of undefined protein added. So when you're adding serum to your media, you don't really know the exact protein constitution of serum. So you don't really know exactly what is in it, molecule for molecule. However, other mixtures like Ham's F10 and F12 were so designed that you can use them to grow cells while adding just the least amount of undefined protein so as to study the effects of the nutrient components of the media. So if your aim is to study the effect of various components of the medium on the cells, then it would be desirable that you know at least most of the components that are present in the media. And under such situation, uh, addition of something like serum whose exact composition you're unaware of will disrupt your studies. So media can be either purchased as prepared liquid media. They can be made up in the laboratory from dried powders, which contains most of the components of the nutrient mixtures. So if you are making media from dried powders in the lab, in that case, you would typically just have to add sodium bicarbonate, which will be a part of your buffering system. And you have to add serum and antibiotics, pen strength. The rest of the components of the medium would already be present in the powder. If you're buying prepared liquid media, that is the best thing you can do, but that is not very cost effective because prepared liquid media would be super expensive. Or media can be prepared in the laboratory from individual stocks of the individual components or groups of the components. So option 3 is the cheapest option, where you straight out prepare the media from scratch. Option 2 is intermediate in terms of cost, because you're buying the dried powders and you're adding some of the components from outside. And option 1 is the most expensive. The major drawback with option 1, other than its cost, is that it would require a lot of space to store, because liquid media would take up a lot of space. On the other hand, if you're buying dried up powders, these will typically come in pouches and you can easily store them. So space taken up for storage will not be too high. And if you're preparing the media in lab from individual stock components, then it is very critical that you pay a lot of attention when preparing media, because even slight variations in these stock components would affect your media quality, and this might lead to inconsistency within the media, which in turn might affect your cell's growth, and therefore not only your cell's growth but their health as well, and therefore the net result of the experiments that you're going to perform with these cells. So you can either buy these powder media, see they come in pouches which are very easy to store, or you can buy bottled liquid media directly. So something that I forgot to mention in the previous slide, there was a link added at the bottom for further reading. So if you go to this link, it will help you better understand what are the various other kinds of media that are commercially available, and what is the advantage of each media, and what are the different medias that are suited for growing different cell types. So that is good for further reading in case you're interested to learn more. So next we are going to talk about serum. So now I briefly mentioned what serum is in the previous slide, and that most of the media like MEM and DMEM, they have been designed to be used with serum supplementation. So what is serum? Now the growth media for most animal cells in culture also includes serum, which serves as a source of polypeptide growth factors that are required to stimulate cell division. So serum is the component of blood that is left behind when you remove the blood cells and the various clotting factors. It includes important polypeptide growth factors that are required to stimulate cell division and are therefore required to promote healthy cell growth. Such growth factors also serve as critical regulators of cell growth and differentiation in multicellular organisms. Most cell lines are routinely cultured in serum supplemented media, and often when performing experiments, say for instance, you would want to bring all of your cells to the same stage of the cell cycle. That is, you would want to synchronize the cells. So one way of achieving this synchronization is to start the cells of serum. So you replace the serum supplemented medium with non-serum supplemented media. That is media that does not contain serum. And this will eventually lead to synchronization of all the cells that are present within your either say petri dish plate or flask. Now, in addition to whole sera, which is allowed to clot and then the clot is removed, blood can also be collected with an anti-clotting agent and the cellular portion can be spun out, resulting in plasma. So, serum is blood minus the blood cells as well as the clotting factors, whereas plasma is blood minus the blood cells. So, it contains the clotting factors. So, you can collect simply plasma. How? You take out the blood. Now, blood when taken out of the body would automatically clot. So, to avoid that, you add the anti-clotting agents and then you centrifuge it to spin and settle down the cellular portion and you remove it. So, whatever supernatant you're left behind with is your plasma. Serum and plasma, even from the same species, are often quite different in their composition and therefore their effect on cells. Culture media can be supplemented with sera from any species of animal. Bovine, fetal, newborn or adult and horse sera are the most frequently used. Most commonly used serum as I have encountered in my research experience is FBS, which is a fetal bovine serum. Most commercial sera comes sterilely packaged, so you do not prepare it. Of course, you do not. You buy these bottles which contain sterilized sera packed in it. Now, serum can be treated before use in one or more ways, that is by either filtration or dialysis or fractionation. So, although they come sterilized, if you feel that somehow the sterility of your serum has been compromised, you can sterilize it by either of these techniques. Most commonly used technique is filtration. Now, these treatments can act as an added insurance against contamination. It can remove or inactivate the toxic components of the serum. It can remove or inactivate growth promoting or differentiating components of the serum and specifically remove low or high molecular weight components of the serum or particular serum fractions. There is anything unwanted that might be present in the serum that you think might interfere with your cell's growth and viability, you can remove them by either of these techniques. Finally, we are going to talk about serum-free cultures. So, like we already discussed, serum is essential for the cell's growth and differentiation. But there are some media which have been so designed that they are to be used without any serum supplementation. Similarly, there are certain cell types which do not require serum supplementation. So, under such scenarios, what one practices is the use of serum-free cultures. Now, serum-free culture involves supplementation of these serum-free medium with defined factors to replace the serum for the growth of established cell lines. So like we discussed a couple of slides back, that Ham's fixtures were designed so as to allow the study of the different components of the media on cell growth. So if you're adding serum, you don't really know what are the various different polypeptide growth factors present in the serum. So it will be difficult for you to study the effect of these individual factors on cell growth. And sometimes you just want to grow your cells in a medium whose 100% composition you are aware of. Under such situations, you're going to resort to the use of serum-free media, and over there you supplement your media with specific defined factors because the cells will be requiring these factors for their growth and differentiation. So you have to supplement the cells with them. If you do not, then you will end up starving your cells. This usually involves the addition of specific hormones and growth factors to the culture media. A number of serum-free media formulations have been described for continuous cell lines as well as for primary cultures. However, switching to serum-free media still needs a time-consuming literature survey and manufacturers search for appropriate media formulations. In fact, switching from any one kind of media to the other kind requires intensive literature survey, and it is very important that you do it to ensure your cell's optimal survival. And also you have to search for appropriate manufacturers who can provide you with the required media formulations, because it is quite possible that these serum free media are not so easily available, or it might be more difficult to source them, so you have to search for their availability. In serum-free culture, the composition and preparation of the medium is far more critical than in serum supplemented culture, because these growth factors will have to be added externally by the manufacturer or by you if you're preparing it yourself. So one has to be very careful and very critical when preparing it, because any slight deviations can actually completely disrupt your cell's growth and survival. Many nutrients commonly provided by serum, for example vitamins, are not available in the simple media like MEM or DMEM, and therefore you cannot use this media without serum supplementation. It is therefore essential to use a more complex mixture like the HAMS mixture that we talked about, or the IFCOS mixture that we talked about, or a mixture of such media in serum-free culture work. So not just one medium, but a mixture of different media. So again, the final media that you're going to use, that has to be optimized by you before use. Now, serum protects against the action of certain substances in media. Media components such as reducing agents can also interact with and inactive the supplements. So you have to be very careful. If you're not using serum and if you're planning on supplementing your media with these growth factors, then you have to ensure that none of your media components interact with these factors and inactivate them, or rather none of those factors interact with your media components and inactivate them or use them up, because ultimately all of these components are required for proper survival and growth of your cells, so you have to be very critical about it. So this brings us to the end of our lecture, which is part 2 of mammalian cell culture. So to briefly give a short recapitulation, we talked about the various conditions for cell culture. We started off with temperature, and we discussed the importance of temperature in maintaining healthy cell culture. Next, we moved on to pH and why it is important to maintain the appropriate pH, what are the different buffering systems that are available to help maintain the pH, and how you have a pH indicator present as a part of your media to help with quick visual assessment of the pH of the cell cultures. Next, we talked about osmolarity of the medium and also the gaseous content. The temperature and the gaseous content is mediated or regulated by the CO2 incubator, and the pH and the osmolarity is regulated by the media composition. The pH is also regulated by the CO2 incubator in case of the bicarbonate CO2 buffering system, where the incubator provides a carbon dioxide component of the buffering system. Next, we talked about different media compositions and what are the various components of media, what are the various uh, components that are required to be present in media that are essential for your cell's survival. Then we moved on to talk about serum. What is serum and what is its utility? We also talked about feeder layers that are required for culturing certain specific cell types. And finally, we discussed about serum-free cultures, what is the utility of serum-free cultures, and how to ensure that your cells survive in absence of serum. So that is it for this particular lecture. In the upcoming lecture on this particular series, we are going to talk about the different culturing practices that we follow and how cells are maintained in cell culture. So we have already looked at the various requirements, and next we are going to discuss about how cell culture is actually done, how to maintain the cells. So in case you have any questions, please feel free to leave it in the comment section and I will definitely get back to you. If you like this video, do subscribe to my channel for more such content. Hit the like button and share this video. So that is it for this time, and I will catch you again next time. And please feel free to leave any doubts that you have. So till then, bye.